Welcome to our presentation on the polymerase chain reaction by Carissa, Kellie and Susan. The polymerase chain reaction is used to make billions of copies of a specific piece of DNA. It is an essential tool in modern molecular biology and has transformed scientific research and diagnostic medicine. The polymerase chain reaction is sometimes called molecular photocopying. The reason being it is a fast and inexpensive technique used to amplify and copy small segments of DNA. Studies of isolated pieces of DNA are virtually impossible without the polymerase chain reaction amplification because significant amounts of a DNA sample are necessary for molecular and genetic analysis. The polymerase chain reaction has been used to successfully perform extracts from whole cells, even in a very old or damaged cell sample, from archaeological samples and degraded samples, especially in the case of crime scenes. The polymerase chain reaction is very important for the identification of criminals and the collection of organic crime scene evidence such as blood, hair, semen, pollen and soil. DNA fingerprints, identification of familial relationships, genomic DNA isolation, and other molecular diagnostics and biochemical analysis can be undertaken forensically through the use of the polymerase chain reaction. The polymerase chain reaction allows DNA to be identified from tiny samples. A single molecule of DNA can be enough to help the polymerase chain reaction's amplification. The polymerase chain reaction is used for a number of applications. Genotyping, which is a process of determining differences in the genetic makeup between one individual's DNA sequence and another. Cloning, which is basically making a copy of a piece of DNA. Mutation detection, we have covered previously. Sequencing is most common in crime scene evidence and archaeological identifications. Microarrays are used to measure the expression levels of large numbers of genes simultaneously, and in forensics, where the polymerase chain reaction can help identify one person from millions of others. In paternity testing, the polymerase chain reaction can also help identify a biological parent. This slide presents the current applications of the polymerase chain reaction, which includes medical applications, forensic application, infectious disease detection and identification, and applications in molecular biology research. The polymerase chain reaction has many different uses in science. The polymerase chain reaction has become an enormous application in any test for the presence of a particular gene or organism, in diagnostics, in medicine, and various other fields like forensics. Biotechnology, the polymerase chain reaction is used routinely to clone genes for producing important proteins in medicine. In medicine, the polymerase chain reaction is used for rapid identification of infectious diseases and the diagnosis of genetic disorders and cancers. The polymerase chain reaction was developed in 1984 by the American biochemist Carrie Mullis. Mullis received the Nobel Prize and the Japan Prize for developing polymerase chain reaction in 1993. The polymerase chain reaction revolutionized molecular biology and any biological science that works with genes.
Significant amounts of DNA are required when analysing DNA in a laboratory. This is why it is essential for the sample pieces of DNA to be amplified by PCR. The first step in the polymerase chain reaction is denaturation. This step requires the following components: the sample of DNA required to be replicated, a heat-stable DNA polymerase enzyme called Taq polymerase, PCR primers, and nucleotides. Each ingredient has a specific and unique task in the process. The process begins with the sample DNA being placed in a test tube and put inside a PCR thermocycler. The thermocycler holds the tubes and is programmed to alter the temperature for each stage of the PCR cycle. The DNA sample is heated to over 90 degrees Celsius until the weak hydrogen bonds between the DNA's nucleotides break. The strength of the heat is responsible for denaturing or separating the DNA into single strands. The next step is called annealing. The thermocycler cools the reaction to a temperature of 55 to 65 degrees Celsius. The synthetic oligonucleotide primers are added at this stage. The primers are made up of a short strand of nucleotides, approximately 20 nucleotides long, that complement the section of the DNA attached by base pairing. The primers attach to the separated DNA strands with one primer used per strand. They bind to the beginning of the sequence of the DNA that complement their coding. This step ends with two separate strands of DNA, both with the primers attached, marking off the specific region to be replicated. The third step is called extension. Once again, the reaction is heated, but this time to only 72 degrees Celsius. The nucleotides begin to add themselves to the DNA strand, beginning at the spot marked by the primers. This continues until the new strand of DNA is identical to the original template DNA. Once done, there will be two identical copies made from the original DNA. Each step in the process is crucial, and maintaining the correct temperatures at each stage is vital. And the cycle continues with each duplicate DNA denaturing, annealing, and extending, making another two strands. Within 30 to 40 cycles, the original DNA sample can make more than 1 billion copies. The cycle starts from one DNA strand replicating into 2, then 4, then 8, 16, 32, and so on. Within a few short hours, the process can generate over 1 billion copies of new DNA. PCR has been essential in the success of modern DNA profiling. This includes human identity testing and crime profiling. It is an incredibly useful tool in helping to solve violent crimes such as a sexual assault or murder, identifying missing people, and examining the remains of victims from mass disasters. The introduction of PCR has had an incredible impact in the field of forensics. Since its inception, PCR has enabled investigators to find answers to unsolved crimes by way of replicating DNA decades old. This can be done by comparing a suspect's blood with a sample found on a victim or by a DNA sample taken from the body to determine identity. PCR is also used in paternity testing, where they test for similarities in DNA to match paternal or maternal relationships. This can be done through the use of DNA found in hair, fingerprints, or blood. Thanks to PCR, testing for paternity is now a quick and easy test, only requiring a swab taken from the inside cheek of the mouth. Prior to PCR, blood profiling was the only scientific method used to identify paternity. It was not a reliable test, as while it could prove someone was not related, it could not prove for certain that they were. PCR is also used in the diagnosis of disease, including genetic, infectious, and autoimmune conditions. PCR is able to detect AIDS, influenza, SARS, and the herpes simplex virus, among many others. From a small initial sample, PCR can generate enough DNA to be analysed in a laboratory.
When looking at the advantages of the polymerase chain reaction, we know that it is most known for its speed and simplicity. The technique is easy to understand and use, and it produces fast results. This means it is easy to train people in the technique, and it is said that the complete beginner can start to perform the polymerase chain reaction experiments and generate meaningful results within a few days at most. The technique is highly sensitive and has potential to produce millions to billions of copies of a specific product for sequencing, cloning, and analysis. The QRT-PCR has the advantage of the quantification of the synthesized product, so it can be used to analyze alterations of gene expression levels in tumors, microbes, among other disease states. 30 cycles of the polymerase chain reaction can increase the DNA sample 1 billion times and can be achieved in under an hour. This produces results much faster than older methods of DNA amplification. The polymerase chain reaction only needs a small amount of genetic material in order to work. The polymerase chain reaction is extremely specific, so it will copy nothing else but the target area along the DNA strand. And this is due to high levels of stringency, which is how good the match is between the primer and its target, and the high temperature of the reaction. The polymerase chain reaction is great for detecting pathogens, and multiple targets can be amplified in a single PCR experiment using multiple primer pairs, and up to 20 pathogens can be covered in one test. So now let's take a look at the limitations or shortcomings of the polymerase chain reaction. So although the PCR has high specificity, it only works under certain conditions. The polymerase chain reaction requires specific sample preparation. Unlike other tests, which can use raw serum blood sample, the PCR requires certain substances to be removed, making the preparation more challenging. The DNA sequence is a factor, and if the length is too long, then it becomes difficult to ensure that the target sequence will be copied accurately and in its entirety. If a primer sequence appears more than once in the DNA molecule, then the primer might attach to more than one area and would cause the experiment to copy a wrong sequence. This leads on to the fact that any form of contamination of the sample can provide misleading results. Because some prior sequence data is required, the polymerase chain reaction can only identify the presence or absence of a known pathogen or gene. The other limitation of the polymerase chain reaction is that incorrect nucleotides can be incorporated into the PCR sequence by the DNA polymerase. This happens at a very low rate. Real-time PCR has arisen as a technological innovation and plays an increasing role in clinical diagnostics and research labs. The future application and development of the technique will see the ease of use and repeatability of detection become the new direction and development of the technology. The polymerase chain reaction technology and sequencing, integrated with automation, will likely become a new requirement for clinical molecular diagnosis and public health emergencies. There are currently academic groups and companies starting to look towards technology to improve the diagnostics of the polymerase chain reaction. And the further development of the polymerase chain reaction will have a big impact on the future growth of precision medicine, with a big picture of enabling customization of healthcare to individuals. The polymerase chain reaction diagnostics will expand into primary care in the future.